Hello, good morning, good afternoon, good evening to all of you joining from different parts of the world today. We have the pleasure of having Dr. Margaret Worthington from the University of Arkansas today talking to us about Blackberry and Muscadine breeding for the fresh industry. Margaret has a lot of roots here in North Carolina. She's a North Carolinian, and she got her bachelor from Duke University and her PhD here at North Carolina State University under Dr. Paul Murphy. She also has a master's from UC Davis, and she spent 1 year as a Fulbright fellow in India in between her bachelor and master's. Margaret is working mainly in blackberries and muscadine breeding, but also she works in peaches and other trees, other fruit trees. So I appreciate Margaret for accepting your invitation. I look forward to your talk, and I'm going to stop sharing my screen and get you to share yours.
Okay. All right, are you seeing it?
Yes, we are. Thank you.
Okay, excellent. Shall I go ahead and get started then?
Yes, you're all set.
Okay, well, thanks very much for the invitation to speak here today. It's a topic that I'm really excited about. You know, when they were asking me what I would like to speak about, I decided that I wanted to talk about two of the fruit crops we work on, blackberries and muscadine grapes. Because I think that these are really interesting for students and maybe other breeders to think about the role that plant breeding can have in building a new industry and crops that are kind of maybe minor crops to start out with and how they can grow that to be a global industry. And I want to talk about the role that the University of Arkansas fruit breeding program has had in the blackberry industry and what I hope our role will be in building the fresh market muscadine industry. So as a little bit of background, let me see, I'm trying to advance my slide here. Here we go. Our fruit breeding program was established in 1964 by Dr. Jim Moore. He was an Arkansas native, but he had gone on to work in Beltsville for the USDA ARS program. And I always get told the story that he was told no one would ever hear from him again if he went back to Arkansas. But he built new fruit breeding programs here from scratch for a number of different crops and built a hugely successful program. So he started programs for strawberries, apples, blueberries, peaches, blackberries, and grapes. And thank God we don't work on all of those anymore because I would go insane. But we do work on peaches, blackberries, and grapes still. And today I'm going to talk about blackberries and grapes and the most newest crop that we started working on, muscadine grapes, in 2007. So a little bit about our program's history. After Jim Moore retired in 1997, Dr. John Clark took over the program. He was the leader of the program from 1997 to 2022 when he retired. And a really nice thing that they did in our program here was they hired me to overlap with John from 2016 to 2022, which is the world's longest overlap, I'm sure. But it was really a great opportunity for me to wrap my head around how our program works and what our germplasm is and learn from his experience. And it worked really well because we do work on so many different fruit crops that we weren't tripping over each other. So I worked on peaches and muscadines at first, and then on dwarf blackberries, and then assumed full leadership in 2022. Another reason it was good we have an overlap is that I didn't have any experience working in fruit crops before I came on here. As Dr. Iglesias said, I did my master's at UC Davis. I did it in bean breeding with Paul Getz, and then I went on to North Carolina State, and I did my PhD in wheat breeding with Paul Murphy, and then I went on to work at CIA on brachiaria forage grasses. So I had no experience working with fruits or horticultural crops at all before I came on here. But I actually think that that was a good thing, especially considering the overlap we had, because I was able to think about some modern things that we were doing in row crop breeding and kind of bring in some of different ideas, I think, to the breeding program here. So I would encourage everybody that you don't have to continue to work in exactly the same crop that you've done your graduate work in. It's kind of cool to go to new different places and work on new different crops to expand your skill set and maybe bring in some ideas across. So, some of the major breeding accomplishments of our program are the development of blackberries that combine erect growth habit, thornlessness, great consumer quality, and shipping ability. Another big advance we made is in the development of the first primocane fruiting blackberries, which is a new category of blackberries, and I'll talk about that a little bit later, but it's been really influential for the industry. Another big accomplishment of our program has been the incorporation of foxy and muscat flavors, and I'll talk about what that is exactly, to develop table grapes with superior texture. And this is something we did in a public-private partnership with International Fruit Genetics to develop products like cotton candy. So we've also made a lot of advances in intellectual property rights and public-private partnerships, breeding agreements, that sort of thing. John has been a real leader in this area for university breeders. So I think we've done a lot of innovative things, and we've been really aggressive in pursuing those partnerships in this program. So as a result, University of Arkansas fruit breeding program products are now grown on all continents except for Antarctica. So look out, penguin, we are coming for you next. Let's see. So, whoops. We have 117 total cultivars that have been commercialized from this program. 65 of those are public cultivars, but we've also done some exclusively licensed cultivars for the home garden market. And we've made a lot of cultivars with breeding agreements with private companies like Driscoll's, IFG, and overall, this has generated in $12 million of royalties that have been collected at least over the lifetime of the program, and about $3 million in agreement income for these breeding and testing agreements. This is really important for our program because you may or may not know about Arkansas, but here it is on the map. Arkansas is not actually like a global dominating force in the world of fruit production. We do have thriving local industries. We've got farmers markets, local sales. We even have a few growers that work with shipper marketers for grocery store sales. But we don't have a growers association that gives us a tremendous amount of money. We don't get any money, in fact, from growers associations to run our program. And what we do is tremendously expensive, growing these high value perennial crops that have to be maintained year after year. So this is how we're really able to sustain our activities, is through these agreement income streams and through revenues from royalties. So where is Arkansas? It may be that not everyone on this call knows where Arkansas is, so I figured I'd highlight it here. We're in the Southeast. The climate is actually very, very similar to where I grew up in Raleigh, North Carolina. It's, humid here. We get hot, humid summers. We get pretty cold though. Usually here we'll get below 10 degrees Fahrenheit every year at some point. And we get a lot of annual rain. We have these hot, humid summers. So I'll say also, it's important to think about for perennial fruit crops, we get at least 1,000 chill hours a year. So those are hours between freezing and 45 degrees Fahrenheit, or about 15 degrees Celsius, I think, that the plant recognizes that it's cold, it's going through dormancy, and this is really important for breeding of fruit crops. So it makes it a little bit more difficult for us to breed for low chill environments because we have all of those chill hours. But anyways, within Arkansas, we work in Clarksville at the Fruit Research Station. So Clarksville is located here. About 95% of our research is there. We have a great staff. It's a beautiful place to work. It is nestled in the foothills of the Ozark Mountains. So it's a nice view when we pull up every morning. It's really a great place to work, and we have a great staff. We have a station director that runs it, and a lot of wonderful staff who have been there for many, many years and are very, very knowledgeable in helping us run the day-to-day of the program. We also have a global footprint for what we do. You know, people will sometimes ask me what the goal of our breeding program is. And of course, I want to grow things that, you know, work for Arkansas growers and growers across the Southeast. And that is my primary goal. But I'm also interested in global domination. So I want our blackberries to be grown in all parts of the world that there possibly could be. And so we have a global footprint working with partners in the private and the public sector on testing in different environments. Those are indicated by the black stars. And we also have breeding agreements with private sector partners that are located in all of these places with the red stars. So this is really important for us in understanding G by E and which of our varieties and advanced materials maybe have a place somewhere else in the world, even if they don't have a place in Arkansas. So another thing that I really like about our crop and our programs here is that we work with crops that are native to the eastern US and the southeast. So blackberries here, you know, I've got this with this red circle. There are blackberries in all parts of the world, right? There are Rubus ulnifolius, this kind of English blackberry that's a very, you know, important species there. There are Rubus arcinus, these western blackberries that are very important in the western US that are mostly hexaploids and higher. You've got Rubus glaucus in South America. That we had a lot of blackberry juice when I was in South America too. So they're all over the world, but the blackberries that we work with in our program are from, they're mostly native species to the southeastern US and the eastern US. So we work with Rubus alleganensis, Rubus argutus, and Rubus trivialis that are all kind of present within this range that I've got highlighted here with the circle. We also work on muscadine grapes, and these are my favorite fruit, and I'll talk about them more later on. Muscadine grapes are native to the southeastern US. They're kind of limited. We're at the very northern part of the range here with our they're limited by cold hardiness, really below 10 degrees Fahrenheit. So where we are breeding is kind of at the furthest extent of how north you would find them. But they really are a dominant understory species in southeastern forests, particularly in areas like coastal North Carolina. So it's fun to be able to work with crops that you can access the wild relatives right where you are. So we're kind of using some of these wild relatives from our backyard. So I want to get into the story of how our program helped build a blackberry industry. So if you were to go to the supermarket store 20 years ago, you wouldn't have found blackberries on the shelf for the most part. Nowadays, if you go into any US supermarket, you will see strawberries, blueberries, raspberries, and in the corner there will be a section for blackberries. And I'm proud of the role that we had, a small role along with a lot of business people, shipper marketers, growers, a lot of factors around putting this together, but plant breeding was really an important part of that. So the history of breeding, you know, people picked wild blackberries for a long time, right? A lot of people tell me that wild blackberries are better than cultivated blackberries they find in the supermarket. They're wrong. It's a false memory. They just enjoyed picking blackberries with their grandparents. The ones that we have bred are, I think, much tastier and easier to pick and grow than the wild ones. But we had started the breeding programs back in like the late 1800s. There was a strong and a thriving processing industry for making jams and jellies and that sort of thing in the western US. But the southeastern industry and California and the fresh market in general has grown a lot recently, and it's kind of become this fourth berry crop. So let's see. Having trouble advancing my slide for a second. Here we go. So yeah, here you can see from 2000 to 2010 when a lot of this really rapid growth happened. And this gray bar here is the Mexican industry. So what really helped set off the growth of the industry is the development of a blackberry industry in Mexico to provide year-round products. And I'll talk about this a little bit more. Another thing I want to highlight is that if you look across all kind of fresh fruits and nuts and their use as commodities, berries in general, and especially blackberries and raspberries, are in the highest growth area. So it's really exciting to see those develop more and more. So what are the factors that have led to the growth of the US blackberry industry? First of all, increased consumer demand, then advanced production methods, improvements in post-harvest handling, and new production regions and general year-round product availability. And I want to talk about the role that better varieties and plant breeding had in all of these first four factors. So in terms of consumer demand, people talk about the health benefits of berries a lot. I do think that that's a part of it for sure, that people know that berries are healthy and blackberries are healthy, and that has been one factor driving the growth. Another factor has been the interest of these shipper marketers and having a basket of berries. They're interested in having another product category that they can have to put in, a beautiful bowl of berries and have on the supermarket shelf. Another factor I think that has led to consumer demand is this better flavor that we have available now. Blackberries are unpredictable, I will say. If I take 5 blackberries and I put them in my mouth, even from the same plant, it is likely that like 2 of them will be great, 1 of them will be fine, and then 2 of them will be super sour. And so we've done a lot of work as breeders to kind of make a blackberry that is more consistently sweet and less tart. They can be tart. So you have reduced acidity, increased sweetness, and also breeding for these volatiles that give it a nice blackberry flavor in the background and for consistency again. So you'll see sometimes nowadays new blackberries being marketed by their name. In Europe, of course, you have to put the name of the variety on the punnet, but in the US, that's not a requirement, and usually they aren't marketed by variety, unlike something like an apple where you're buying a Gala or you're buying a Cosmic Crisp or whatever. People don't know what blackberry their variety they're buying for the most part. But now there's a new variety out in Mexico called Sweet Caroline that's very, very sweet, that's being marketed by its name. You see Driscoll's coming out with something like the sweetest batch category to differentiate. We have a new variety called Sweet Art Ponca from our program that is really, really tasty. And I know that at farmers markets, a lot of growers are marketing it by name because consumers want to come back and get it again and again. So improving the taste, to me, that's much more important than breeding for health benefits in terms of like generating new market. If it doesn't taste good, no one's going to want to buy it ultimately. So another development is post-harvest handling. Blackberries are one of the most perishable crops you can deal with, period. Like they really don't last very long. So having a good cold chain, having the infrastructure to get these materials overnight from the areas of blackberry production in Michoacan and Jalisco up to the US market in 24 hours, these things are important. But breeding has also played a role. We've bred very hard for firmness in our breeding program and in other fresh market blackberry breeding programs. We have issues like leak that are associated with less firm berries. And another issue that you see often that is a big problem for growers is red druplet reversion. So you'll pick a blackberry and it looks fully black and ready to go. But then after you put it into cold storage, drive it on a truck for a while, pull it out, then you pull it out of the supermarket and it gets these red drupelets that have come back to a red color because of anthocyanin degradation. And what we learned, through studies here and elsewhere is that it really seems to be membranes that are broken either through mechanical damage during picking or shipping, or just the shock of going from hot to cold. And then you have those anthocyanins that leak out and they revert to their red color. It doesn't actually affect the flavor of the blackberries at all, but consumers look at this and they read it as unripe, even though it really isn't unripe. So it can result in reduction of shipments, and it's something that consumers don't want. So we are breeding hard for a firm blackberry that can be picked and shipped without reverting to red and leaking all over the place. Advanced production methods. So caneberries traditionally are very thorny, as you guys may know. They are labor intensive. First of all, I'll say they have to be handpicked. There is no viable way to machine harvest fresh market blackberries today, nor for the foreseeable future, I think. They're really tender berry and they need to be handpicked. So labor is an issue. But we've been able to breed them to be thornless. That makes management of the plants and I think the picking much more appealing. In the US, I don't, being thorny is a killer for a new variety in the US and the European market. In Mexico, they are tolerant of thorniness in a way that we are not here, but I expect that that'll probably change in the next 10 years. So we've bred for thornlessness, and I would say now 95% of our material in the Arkansas program is thornless. We bred for erect growth habit as well. Here you can see blackberries can be trailing, they can be kind of semi-erect, or they can be erect. We've bred really hard for erect growth habit. Most commercial growers, I mean, any commercial grower really is going to use a trellis at this point, but having a blackberry with an erect growth habit still makes it easier to manage. You can see you have to wrap canes like this. If you're working with like a trailing type blackberry, like these processing kinds, here, we've bred for a blackberry that will stand up on its own and you're really just using the trellis to keep it in its place. Reduced stature. This is an area that we are working on a lot now. Blackberry is kind of a gangly plant. If you don't tip it or do any kind of summer pruning, a blackberry plant will get to be like this 10-foot tall monster. But we have, within our program, a lot of variation within growth habit. We have new varieties like Sweetheart Ponca that are a full-size plant but with reduced internodes. And we also have within our material some really dwarfy material. We're working on developing that for the home garden market. We're working with home garden companies on making kind of an edible ornamental blackberry, and they are very cute. Here I am laying by a really short one. We also think there might be potential applications for this for, either vertical farming indoors or glass house production, like how you have strawberries that are grown on tables in Europe. So working on increasing the harvest index and making this a more manageable plant size is another area. So the last thing I want to talk about is new production regions and product availability year-round. A thing about fresh fruit is if you do not have year-round product supply, you will lose your spot in the supermarket. You absolutely have to be able to, 54 weeks of the year, provide fruit to fill it up. So the growth of the Mexican industry has been hugely important and I think positive for most US growers, particularly because the rainy season in Mexico, where blackberries are primarily grown here in Jalisco and Michoacan, is their rainy season is like May through September. It's exactly coincides when fruit production would come on in the US. There's some overlap that might make it difficult for Florida growers or maybe South Georgia growers, but for Arkansas growers, they're absolutely not competing with the Mexican market. And instead, the Mexican market is providing product to keep something on the shelf. So, the Mexican industry in 1995 was only 230 hectares. By 2015, it was over 5,000 hectares. I'm sure it's more than that by now. The development of that industry was dependent upon the availability of low chill varieties. So varieties that don't require that same number of hours under 45 degrees Fahrenheit or 15 degrees Celsius or whatever. It doesn't have to be that cold for them to produce. There are ways that Mexican growers have come to trick low chill plants into thinking they've got chill hours, even if they have less than 50 chill hours that occur, which is the case in a lot of these production areas. What they do is that they apply hydrogen cyanamide, which is kind of like a Dormex, kind of to burn the leaves off. And then they come back with Revent or kind of a cytokinin mimic to increase growth after that. So it's a kind of recipe that they've developed for working with this variety Tupi, that is a low chill variety that's traditionally been dominant in Mexico. Many of us are looking to come up with replacements. Another area that has really helped with the growth of the Mexican industry and global production in different environments is the development of primocane fruiting. So let me talk about what that is. So primocane fruiting, blackberries traditionally, I guess, are they're a perennial plant, but all of the canes are biennial. So they're unique this way. So a cane comes up in the first year and it will be vegetative that whole first season. Then it goes through dormancy, comes out in the spring, breaks bud, makes leaves, and then flowers in April. Now we're at kind of peak blackberry flowering here in Arkansas, and the fruit will come on mostly in June here, okay? Now, a primocane fruiter will do what the floricane fruiter traditionally does also, but those first year plants, once those first year canes come up, they will start to they'll start to flower maybe in May or June, a little bit later. And they'll continue flowering through the summer, depending on what your climate is like. And you can get a fall crop. So it extends the harvest season. So it's something that's interesting to growers in Western North Carolina, growers in California, and temperate parts of the world, because you get a different season. You get this kind of late summer fall crop of blackberries that can complement your main crop from the floricanes and come in at a time when you can command a higher price. The primocane fruiting blackberries are also, I think, really interesting for different environments where you may not have any chill requirement. There's no chilling involved at all. So they'll produce even in an environment with no chill hours. It might also be interesting, and we still need to work on improving this, is for areas where it's too cold to grow blackberries traditionally, where they would have too much winter injury to the canes, while they're overwintering before they can fruit and flower. So if we can make one that comes up and primocanes fruits early enough, then maybe it's something that they can grow just for the primocane crop in areas like New York. So this is a technology that was developed here at the University of Arkansas, and I want to talk about why it's important that we have this global footprint now, because most primocane fruit in Arkansas does not look good. We have really hot summers, and the pollen viability of blackberries goes down significantly when you get over like 90 degrees Fahrenheit. What's that, 35 degrees Celsius, something like that. It gets When it's that hot during flowering, we get pore set and we end up with these kind of small misshapen fruits that are typical here. So we're able to select and improve on primocane fruiting, but this isn't the environment that it works the best in. Where we really discovered the value of the primocane fruiting trait initially was by working with our colleague Chad Finn in the ARS program in Oregon. That's where John discovered, the same variety might look beautiful and yield so much better where they have a milder summer. So that's one thing to keep in mind. It's important to breed in these different environments. So now I want to shift gears to talking about kind of the future of blackberry breeding and what we're doing to make our program more efficient and keep up with the times. I'll say I have a significant interest in our program being efficient because I have 3 very small children between the ages of 4 and 4 months. And so I have a lot of work to do to go down to the field, make it down there. It's a 3-hour round trip drive every day in the summer, make the selections that we need, do a good job, make sure we're competing with the private sector, we're getting the varieties out there to the places they need to be. And I definitely am interested in using the science side of breeding in addition to the art, which is what's really driven our program in the past. So, when I started, we had basically no genomic tools. We were doing all our data collection in notebooks, and we had a great breeding program, but we had a lot that we could improve on to use new genomic tools that were available and to improve the efficiency of our data collection and analysis. So the first thing that I did when I started with my startup was I thought, well, okay, we need a reference genome. We don't have any kind of reference genome. So I kind of worked together with a lot of partners to build this big team to develop the first Blackberry reference genome assembly for Hillquist, which is a diploid relative of Blackberry that's actually the original source of our primocane fruiting trait. So we have a nice chromosome scale assembly that we can use, which has been hugely helpful to me in my program. We also started out doing a big genome-wide association study. So we took all of the material within our breeding program and We came up with ways to genotype it, and we phenotyped it for a bunch of different traits. We were complicated somewhat by the fact that blackberries that we breed are autotetraploids. Fortunately, we can build on a lot of interesting work that the potato breeding community and also the blueberry breeding community have done. And we worked with this Tools for Polyploids project. We have used capture-seq markers, kind of following what the University of Florida Blueberry program has done. And we are able to estimate both the additive effects of these different markers with different allele dosages and dominance effects and run this GWAS. And it's been, I mean, I was hoping it would be effective, and it has exceeded my expectations for what we've been able to discover about our program using this approach, really without taking too many resources away from our applied breeding activities. So we have phenotyped about 400 Arkansas breeding selections for a bunch of different traits, including thornlessness and primocane fruiting, that kind of are more inherited as qualitative or major effect allele traits. And then also fruit texture, red druplet reversion, internode length for our plant height, sweetness, acidity, seediness, berry size, shape. And we found interesting results for a lot of these that are, we're still kind of working on pushing out a lot of these papers. But let me see, I want to highlight here the results we have for primocane fruiting. So up to now, no one had found any kind of genetic locus that really was explaining the variation in primocane fruiting in any meaningful way for raspberries or blackberries. Using this GWAS approach, we were able to find a peak marker on chromosome 3 that, had an LOD of 80. It really explains a lot of the variation in primocane fruiting. And based on this, we designed a molecular marker. So we run this molecular marker. We ran it on 3 populations in 2023. Now we're running it on 5 segregating populations. Why this is important for us is because of these polyploid segregation ratios, we ended up with, if we did a cross between something that's primocane fruiting, so it has 4 recessive primocane fruiting alleles, and something that is floricane fruiting with half and half. So it's got 1 primocane parent. Anyway, it ends up with 2 primocane alleles and 2 floricane alleles. We would end up with only 1/6 of the progeny from that being primocane fruiting. We really are interested in improving our primocane material. Our fruit quality, the flavor on the primocane material lags behind what we have in the floricane material. So it benefits us to be able to make crosses between these 2 pools that we're working on, and for us to be able to enrich those population numbers so that we're able to say, okay, actually now all or most of the material that we have from these crosses between floricane and primocane are going to be primocane, and we're going to be able to advance that program. So we have developed this molecular marker, and we're running marker-assisted selection in-house now and throwing out seedlings that are floricane fruiting to enrich the primocane fruiting percentage in these populations. And I think it's been really successful, and it's a nice approach that we intend to move forward with. We also have some ongoing research. We're doing kind of a metabolomic and genomic selection approach to improve our ability to breed for better flavor. Currently it's just me standing out there eating fruit and being like a 7 on a 1 to 9 scale. So we think that using some consumer panels and GCMS data and kind of putting this data together, again, kind of following an approach that had been pioneered by tomato and fruit breeders at the University of Florida, that we'll be able to breed for better flavor in blackberries. We also are doing research on fusarium, which is a major issue threatening growers in North Carolina and now Arkansas. We're working on fruit firmness and red druplet reversion still, primocane yield components, and how we can select for low chill or adaptation to these low chill environments, even though we are working in a high chill environment. And I'll say here too, I put this big, freighter ship on here that this is a big program, and we do want to make changes with the time, but I also have a lot of staff that I'm working with, and I think you don't want to throw the baby out with the bathwater. It's a big ship to steer. So we're making changes slowly and keeping everyone comfortable with doing digital data collection using databases, etc. So enough about blackberries. Now I'm going to shift on to a story about muscadines, and grapes, okay? So we've had a grape breeding program since 1964 here, and we've had a number of releases that are really nice for the fresh market with great flavor. But ultimately, you know, the industry for table grapes is highly concentrated in Southern California. Okay, so our goal was to build an Eastern table grape industry, but it rains here. It's really hard to grow grapes competitively, economically for growers in an environment when it rains. They're susceptible to many diseases. They crack in the rain. So it's been kind of a struggle. And where we've made our major impact in the industry and our grape breeding program has been through our collaboration with the private sector. So John worked with David Kane, an old friend of his and a breeder at IFG, a new company that he started, to send out a lot of our material that was highly flavored there to develop some new grapes. Now, at that time, the conventional wisdom was that consumers wanted sweet, neutral grapes, okay? But our program had a lot of highly flavored material. Where did this flavor come from? Well, with the crunchy table grape texture, of course, comes from Vitis vinifera, which is the wine on the table grape that is grown, you know, across the world for lots of different applications, right? But it's also the source of where we got muscat flavor. Muscat mostly comes from a compound called linalool. You see it mostly in wine grapes. So you'll have muscat wines that are a nice fruity flavor. We also combine that with foxy flavors from Vitis labrusca mostly. Vitis labrusca is an American grape species native to the northeastern US. It's what is used to make Welch's grape juice and also Manischewitz wine. And so it has this flavor that comes from a compound called methylanthranilite that people will call foxy. A lot of people talk about foxy as a bad thing, which I think it probably mostly is in wine, but I love a fresh market foxy grape. So we have worked hard to incorporate all these flavors together to make a really nice, intense experience in eating a grape that's different than just like a neutral, sweet Thompson seedless. And it actually has turned out to be wildly successful. Cotton candy has been probably the #1 income generating product that's ever been developed with this program in Arkansas. Jack Pandal of Grapery actually put his email address on bags of cotton candy grapes, and they have this, they taste like cotton candy. For anyone who hasn't tried them, you know, they have this kind of burnt sugar flavor. I think they're really, really good. He was worried that the flavor would be too much, or that the berries would be too small, or maybe too yellow. They're a little bit of kind of a golden thing, but no one has commented on berry size. No one's commented on the shader color. He says that the comments he get are all about the eating experience, the texture, and the flavor together, okay? So this brings us back to our goal of creating an Eastern table grape industry, right? Like, ultimately, you know, on the table grape side, we kind of decided if you can't beat them, join them. We'll work with the Californian people, but we still want to develop an industry for people in the part of the world that we're working in. And that's why we shifted our focus also to muscadine grapes, the original Eastern table grape. So muscadine grapes are a distant cousin and it's a different subgenus from Vitis grapes, okay? It's in subgenus Muscadinia. They have a different chromosome number from bunch grapes. Basically what we discovered in this program through making the first linkage maps is that chromosome 7 of bunch grapes segregates as 2 independent chromosomes in muscadine, so chromosome 7 and chromosome 20. They are the first cultivated grape in North America. Here you can see the mother vine of Scuppernong, and that's in Manteo, North Carolina, and it's the original kind of bronze or white fruited muscadine. So the vine is still here in existence, grown by North Americans, by the original colonists that settled, European colonists that settled to Eastern North America as well. Let's see, they are a nice big fruit. Here I am holding them. There's a lot of variation in size and color that we have. Did I have on here? I think I missed where they're adapted to. They are native to the southeastern US. As I said, they're limited by their cold hardiness, but if you were to go to the forest in North Carolina and look in the understory, the dominant plant that you would see in the understory is little muscadine grape seedlings. They are native to the southeastern US and they are adapted to our hot, humid climate. They do things like not breaking bud until late April or May because they know about false springs that are common in the southeastern US. They're a really nice, well-adapted crop here, and they're resistant to most of the major diseases that plague grape production around the world. So they have a unique fruity aroma. My car always smells amazing when I take Muscadine grapes back. And they have this intense flavor, but they also have a thick leathery skin and gummy flesh with large bitter seeds. So the majority of muscadines produced in the southeastern US in our industry here are for wine production. And I'm pro wine production, but I am especially pro fresh market muscadine grapes. I grew up eating them, every September. You drive down to the beach and they would be selling them on the roadside stands. Or, you go to the North Carolina farmers market and they'd be there. They were kind of a seasonal specialty crop. And I think they are the #1 best tasting fruit ever. So people may or may not agree with me. It's a polarizing opinion. But the broad appeal is limited by the texture. So they have, again, this kind of thick skin and this like kind of gelatinous flesh. So our goal in the program has been to breed muscadines to be an inside fruit. Currently, muscadine eating involves a lot of spitting. It is fun, but not very sophisticated. So here I've got a picture of a rocking chair and a little spittoon here where you might spit out your seeds and your skins traditionally. To appeal to new consumers in different parts of the world, different parts of the US, cultivars have to have thin skins and they have to have crisp flesh. What we have learned from our experience with cotton candy is that people are excited for new flavors as long as they don't come in a texture that is off-putting to them. So the skin has to be edible. Skins of muscadines can be like shoe leather, but we have new cultivars that have really nice edible skins. I think a lot of the modern cultivars, I eat the skins for sure, and I know that we have even better germplasm in the pipeline. The flesh texture, I've had grad students tell me it's oyster-like, snot-like, and also eyeball-like, so they'd just be flying off the shelves, right? But we actually have within muscadine germplasm material that is much more similar to a table grape, kind of a firm, meaty flesh adherent to the skin. So that material is all there. It's available. The last hurdle is seedlessness. Muscadine seeds are huge and bitter and unpleasant, and there's no way around it. There have been attempts to bring stenospermocarpic seedlessness to muscadine grapes many times over the years. Stenospermocarpy is where the grapes actually, the seed gets fertilized, it starts developing, and then that development is arrested or aborted during the middle of the process, and you end up with this seed trace. So it's somewhat noticeable sometimes, but it's not like a hardened seed. A stenospermocarpy is great actually because that developing embryo brings on the hormones to make a nice big fruit before that development stops. So you end up with a much bigger fruit than you would like a parthenocarpic seedless plant. So there have been many efforts to bring in disease resistance from muscadine to vitis and seedlessness from vitis to muscadinia. And people attempted to make synthetic tetraploids, to use cisgenic approaches, all kinds of things. Ultimately, a private breeder named Jeff Bloodworth, who works in Hillsborough, North Carolina, just brought it in through throwing enough pollen at the problem, making these wide crosses with these kind of bridge hybrids with restored fertility to get over this sterility that you have when you make this initial cross. Crossing a muscadine and a grape is a little bit like crossing a donkey and a horse. Muscadine has the higher chromosome number and thus is more like the noble horse, and grape the lowly donkey. But you end up with a mule that is sterile. So we don't have the ethical considerations that you have with trying to breed a mule to make it not sterile. And we have a lot of pollen and a lot of plants and seeds that we can throw at this problem. So we have developed now muscadines with stenospermocarpy. Jeff Bloodworth and the Gardens Alive program have released 2 cultivars, Razzmatazz and Oh My, that are seedless now, and we have developed a breeding agreement with them where we've been crossing with that material since 2017, and now we have 34 seedless selections in our program that are moving through. I'm really interested and passionate about working with Vitis Muscadinea crosses on both sides to bring in the disease resistance and the quality. We have used some initial marker work to kind of be able to develop some chromosome painting to look at like which chromosome segments are inherited in these wide crosses. And we have worked a lot with the Cornell team and USDA-RS from the Vitis Gen projects on this area. Some other things we've done, we have mapped loci for sex and berry color, which the bronze berry color actually is differently inherited. It's a GST relative to the MIB gene on chromosome 2 that controls berry color in grapes. And we just recently have worked with Cornell scientists to validate that markers for both sex and seedlessness seem to be predictive broadly in both Vitis grapes and Muscadine grapes. So it's a really fun area to work in because we can use a lot of the tools that have been developed by Vitis vinifera breeders and other Vitis breeders and apply those to Muscadine grapes despite the genetic distance between the species and do a lot of comparative genomics to discover cool things that are mutually beneficial, I think. So some challenges that we have for muscadines, bringing this to be a new industry in the way that I think it totally is justifiable to be, because it's a great product to kind of make this the next Blackberry. That I will say is my goal. When John Clark started the breeding program or started working in the breeding program here, my predecessor, blackberries weren't on the supermarket shelf. Now it's a global commodity. I would love for fresh market muscadines to be the same by the time that I retire. This is my reason for coming to work in the morning. I think we have a lot of questions though about who's going to grow them and how to market them. Some people think muscadine has a negative association because people are thinking about that weird texture and muscadine wines that they maybe don't like. So Nature Ripe has grown some of the razzmatazz grapes, and they're actually marketing them as vine drops. Another thing I think is, are they just grown within the Southeast at first, or do we want to target new markets? They've been really, really popular and successful in Asian markets. Also, do you want to call it a grape or a berry? So last year we had a big meeting of the North American grape breeders here. And afterwards I sent back with a berry shipper marketer and a major grape grower shipper marketer vertically integrated company, right? I sent them to both. And the results that I got from like consumer feedback with the berry growers were so much more positive than the grapes. I think the grapes, they are expecting to get one thing and it's different. It's a different experience. And I think it's off-putting to them where like if you think you're going to eat a grape and you eat a muscadine, you'll be like, what is this? Whereas if you kind of make it a new product category, maybe a fifth berry, then I think it could be more interesting to a broader group of people. We also hope who will grow them. I mean, would a traditional wine grape grower or a muscadine grower who's used to the processing industry be better? Or as this industry grows, I think it's a real opportunity for berry growers, actually. So somebody who grows blueberries or blackberries in the southeastern US, like you're used to dealing with the fresh market, you're used to dealing with these shipper marketers. It's a fall crop, so it's going to come on in September, August, October, instead of in June, July, when you're going to be busy with your first crop. So why not do this to kind of spread out your labor across the season and have a secondary source of income? How will you grow them? Is it the same with these hybrids as it is with the other ones? A lot of things need to be worked on and quality standards to really differentiate these new muscadines from the old school muscadines. And the last thing I think is product availability, that it's never going to become a commodity that has a place on supermarket shelves outside of Eastern North Carolina if it doesn't have product availability year round. So is this something that could be grown in Brazil? Is this something that could be grown in Mexico? There are lots of environments where I think muscadines could be grown, particularly because they are a low chill crop. So we need to work with people across different parts of the world. And I think in our program, we're in a nice position to be able to help grow this industry because of our existing connections with the berry industry to help get the word out about these grapes and work with growers to figure out how to grow them better. So anyway, it's an area that I'm really excited about. And with the last thing I'll say is talking about the domestication syndrome and new crops. I think it's fun to think about. I started working with Paul Gepps at UC Davis, who does a lot of work on domestication when I did my master's. And now I can see that we're working on a lot of these same domestication traits that have been worked on with a lot of the older crops with these modern new crops, things like seedlessness, perfect flowers, thornlessness, reduced plant stature, all these things. So that's kind of a fun area. Well, with that, I'll just end and say thank you, and I will take any questions from the audience about the presentation today. So thanks very much.
Thank you, Margaret. Appreciate very nice presentation. We can sense your enthusiasm, and that's hopefully is contagious to a lot of people in the audience. I'm amazed on how can you can develop such competitive programs in species that are not prevalent in your state and become a powerhouse. That's amazing. Okay, that tells us how things can be done in spite of not being a major crops. Okay, so we have a few questions already in the chat. I'm gonna start with a couple of questions coming from Wagner Benedito from West Virginia University. In the quest for higher sweetness, are we sacrificing nutritional value? That's the first question. And the other one is the thornless, what's the genetics of it?
Okay, so first of all, on the sweetness issue, well, okay, I guess you will be eating more sugar if they are sweeter. So that's for sure the case. But I think in the case of blackberries, by breeding them to be less sweet, it's not like we're breeding them also to have lower anthocyanin content. In fact, I would love to breed blackberries to have higher anthocyanin content because I think they'd get less red druplet reversion. To me, for fruit crops, okay, I just don't buy it. That I don't think that it's that useful an effort to breed that hard on nutraceutical quality. Fruits are good for you, for sure. If I make a blackberry that tastes bad, I don't care if I've bred it to be healthier. Fewer people are going to eat it. By making this a product that is appealing to consumers, in addition to being healthy, I think people will ultimately benefit more. I think the same thing could be said for muscadines. Muscadines get a lot of positive press for their health benefits. I am sure that by breeding a muscadine to be seedless, I am making its nutritional value lower. That being said, nobody eats the seed. You'll spit it out, so it doesn't ultimately matter for consumers, and people will still grow them that they can make a vitamin nutraceutical with them. I want to make a product that people are excited about and people eat. And through that, I think we will increase nutritional content. Okay. Now,
thornless, yeah.
Thornlessness. So there are multiple sources of thornlessness in blackberries. There's like these kind of paraclinal chimeras like Lincoln Logan that are dominantly inherited and so on and so forth. But we work with a specific source of thornlessness from the cultivar Merton thornless. It was developed originally in England, and then that material was brought over here. It comes from a wild mutant thornless Rubus ulmifolius. It's actually really interesting because when we do the GWAS, we see this huge peak around the thornless locus. And then we actually looked at like LD decay across our genome, and we have a huge block that is basically fixed from how hard we selected for thornlessness. So I think that is an area for us to tackle, is how can, basically 1/28 of our genome is just fixed with very little variability because we selected so hard for thornlessness. So it'll be an interesting challenge for us to think about how we can maybe bring back some of the diversity around that we've lost with selecting so hard for that domestication trait.
Question here from Sue. Is there a way to reduce the tendency to create a bramble through breeding home garden varieties besides pruning for the inattentive gardener?
Wait, is brown rot?
I don't know. I don't know if Sue, you can text with a clarification. In the meantime, we can
brown
go to
rot.
the next.
Brown rot is a
bramble.
bramble. Well,
Bramble.
bramble is a word that is used for describing blackberries and raspberries sometimes.
Yeah.
but I don't understand the question.
Okay, so let's go to the next question while we wait for clarification. Climate change, what effects has in your targets and for breeding in the coming future? I mean, in the case of these berries and grapes.
Well, I would be hoping, we are kind of are working on muscadines at the very limits of how far north you can breed them and how cold it can be. And I would be hoping that climate change would help me out by making it less cold here, but it hasn't worked out that way recently. It seems like it gets plenty cold here in the winter, and maybe that's the more extreme events that you get with climate change. For us, we definitely are interested in breeding for heat tolerance in blackberries. That is a big area of interest for us. We have a huge amount of heat problems. So we definitely are selecting for things that produce berries, even when it's quite hot outside. It rains here. I would think that we are in such a god-awful environment for blackberry production here with how hot and rainy it is that we will be helping other places to be adapted for climate change because maybe they have conditions like Arkansas coming their way.
Question about ploidy level. Have you encountered significant issues related to the ploidy as you pursue market-assisted selection, particularly with regards with diploid reference genomes?
Ploidy certainly is a challenge with molecular breeding. I think I got accustomed to working with weird polyploids when I worked at SIA on Brachyarea. Brachyarea is like a weird segmental allopolyploid, and so I got comfortable with the messiness of it. We have to have really good marker depth of coverage to be able to call allele dosage accurately. That's, I think, the most challenging part of it is we have to spend so much on our genotyping to get really good data back, because if we don't, if we get like just GBS data or kind of like low coverage, it's basically worthless to us. To me, the diploid genome has been really, really useful. I know that in the University of Florida, they're coming up with a phased tetraploid genome. I'm honestly not sure if it's going to be that much more useful for us than the Argutus is, because it is so closely related to our tetraploid genome. It's been really, really helpful for us. But the segregation ratios and the difficulty of getting good genotyping information are the major challenges. But again, we're working on the Tools for Polyploids project with a lot of co-PIs like the Sweet Potato Program from North Carolina State, and Xiaobang Zhang and Marcelo Molinari are involved with that. So we try and take advantage of new technologies that have been developed.
That's great. That's great. Somebody asked a question, how often you accept students for graduate studies, and imagine they can contact you or.
Yeah, for sure. I think I've got my email on the bottom here.
Your email
send me
is
an
there,
email if you're interested. Everyone has different numbers of students that they are comfortable with. I like to have around 4 at a time. And
4 at a time.
And I've got a number of grants in right now, and so I'll probably be recruiting a student or two in the next year. So send me a message if you're interested. I think it's a great place. If you're interested in doing applied cultivar development, if you're interested in learning about the business aspects of breeding and working with the private sector, this is a great place to come and do it.
That's amazing. A lot of compliments in the chat about your presentation, your enthusiasm, and amazing. Okay. Thank you so much, Margaret. Thank you for everybody that has attended the seminar. We look forward to having you next Thursday when we have Dr. Patrick Cumby from Arborgen talking about making genetic improvement in forest trees. Thanks, Brandon and Tiago, for your help. You guys have an excellent weekend, and we'll see you next week. Thank you, Margaret.
Thank you.
Okay, bye-bye.